Hey Honors Bio, Mr. P here. Today's topic is lipids, and if you recall from last lecture, we talked about carbohydrates. Just a quick review, remember that the monomer of carbohydrates is monosaccharide, the dimer of carbohydrates is disaccharide, and the polymer of carbohydrates is polysaccharide. Okay, how are those monosaccharides linked together? That would be dehydration synthesis. Okay, remember we are removing water to free up bond locations, okay, or bond electrons so that we can link two monosaccharides together. And how is it that we split a disaccharide into two monosaccharides? That would be hydrolysis. Okay, remember hydrolysis is all about adding water, splitting that water molecule into the hydrogen and hydroxide so that we break a bond that holds the two monosaccharides together. Okay, but let's talk a little bit about lipids. Lipids are found in virtually everything that's living, and it could be things like fats, which are found in animals. It could be things like oils, which are the same triglyceride type lipid that a fat is composed of, but it's found in plants. Okay, oils are like plant fats. Waxes, cholesterol, and steroids are the other types of triglyceride type lipids, and then you have phospholipids. Okay, so the two main classes of lipids would be these things that are triglycerides, which are fat, oil, waxy, cholesterol in nature, and then you have phospholipids, which are the molecules that make up all of the biological membranes. Okay, cell membranes of both of prokaryotes and eukaryotes. Okay, we talked about carbohydrates being a high energy or high energy short-term energy storage molecule. Remember, you eat food, you eat carbohydrates, you are going to store that energy in your cells as short-term energy storage. Okay, we cannot store energy for very long periods of time in the form of carbohydrates. So how is it that we convert these short-term energy storage molecules into long-term energy storage? We change the chemical composition of a carbohydrate into a lipid so that we can store this energy long terms in the form of fat if you're an animal or oils if you're a plant. Okay, so triglycerides are made up of these two components. One of them would be this glycerol molecule, and notice that C3H8O3 looks like C6H12O6, but you'll notice that it's not a 1 to 2 to 1 ratio. Okay, we talked about in class that all carbohydrates are carbon, hydrogen, and oxygen, and they're found in a 1 to 2 to 1 ratio. And if it's carbon, hydrogen, oxygen, and it's not a 1 to 2 to 1 ratio, then it's a good chance it's a lipid. Okay, so they have this glycerol head, and then they have this fatty acid chains. And if you see a molecule that looks like this, that's a triglyceride. Okay, tri means 3, glyceride means glycerol, and so you have this glyceride or glycerol head, which is here, and you have the 3 for tri, fatty acid chains. Okay, so these are the fatty acid chains. The glycerol head would be here, and when you put a glycerol head on 3 fatty acid chains, you get a molecule that is named triglyceride. Okay, 2 components, 3 fatty acid chains and 1 glycerol head. Where is all the energy stored? All the energy is down here in the tails. Okay, all the energy is stored in the tails. The head is just kind of the base that holds or supports the tails. Okay, but energy is down here in the tails. If we go into this triglyceride a little further, you'll see right here, which is the box I just drew, okay, that is our glycerol, and it's C3H8O3, okay, just like we had before. And if you counted all those molecules up, or all those atoms up, you would see that carbon, hydrogen, oxygen is in a basically 3 to 8 to 3 ratio. And how is it that we add one of these fatty acid chains onto our glycerol head? And this should look familiar. We take a hydrogen from the glycerol, and we take an OH from our fatty acid chain, and when you add those together, H and H means 2H, and O is O, and so we get H2O. This is a water molecule, And so this process right here is dehydration synthesis, right? We are dehydrating this molecule by removing a water molecule. And when we remove a water molecule, we can then bond this oxygen to the carbon to link this fatty acid chain to our glycerol head, and that would happen a total of 3 times. Okay. You'll notice that this fatty acid chain is completely saturated with hydrogen. Okay, and we call these saturated fats. You've probably heard in health or nutrition classes that saturated fats are bad for you. Why are they bad for you? Because they're solid at room temp. And if they're solid at room temp, which is like 72 degrees, chances are they're going to be solid at body temp, which isn't that much hotter. It's more like 98 degrees, right? And so, has everybody cooked hamburger or bacon and what was solid fat on the meat at refrigerated temperatures all of a sudden becomes liquid when you're cooking it, when you raise the temperature closer to like 150 to 200 degrees? Well, if you let that grease sit around for a while, what happens to it? It solidifies, okay? And so if it solidifies at room temp, then chances are when you eat large amounts of that saturated fat, it's going to solidify at body temp. And if it's solidifying at body temp, that means it's going to potentially clog arteries, it's going to potentially clog cells, it's going to lead to a lot of bad side effects. Okay? Animal fat isn't terrible for you, but you need to keep the saturated fats to smaller amounts, right? And so when we talk about fats being saturated, what are we actually talking about? The answer is we're talking about it being saturated with hydrogen. Notice that every carbon here and here and here and here and here and here all have H2 attached to them, okay? Meaning that we are completely saturated with hydrogens all the way through the chain. And so when all these hydrogens are completely loaded to the carbons, or the carbons are completely loaded with H, they are said to be very inflexible. This chain is very rigid. You can't flex this chain. It's not bendable. It's not pliable. And if it's rigid, which it is, that's why it leads to a solid structure, right? Something that's not very rigid or very pliable is going to remain solid. Okay, so what can we do to make these chains more flexible? And if they're more flexible, then they're more liquid at room temp. And if they're more liquid at room temp, then they're not as bad for the body. We call these unsaturated fats. Okay, unsaturated fats, meaning that they are not saturated with hydrogen. And you can see this kink right here. Okay, what's the difference? You have these CH2 and CH2, 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 all of a sudden CH1, CH1, CH2, CH2. Well, what happened when you removed 2 of the hydrogens from this tail? It created this double bond right here, okay? And the more double bonds you have, the more flexible it gets, because we now don't have all that hydrogen that are keeping this chain really rigid, really solid. Okay? The more double bonds we have, the more flexible it gets. The more flexible it gets, the more liquid it stays. These are the things that are more plant-based, plant oils. Okay? You don't see a plant that has a large amount of fat reserves, because plants don't need to store much long-term energy, because where do the plants get their sun or get their energy? They get all their energy from the sun, and the sun, as far as I'm concerned, isn't going to stop shining anytime soon, and the plant knows that. And if the plant knows or assumes that the sun's not going to stop shining, it doesn't need to store a lot of energy for the long term. And so there's less energy in the liquid plant oils, and that's just because the plants don't have to store a lot of energy, because again, the sun will always come out. Okay, yes, there's going to be days when it's cloudy and the UV index isn't as high, or the amount of light isn't as strong, and so the plants need to store some energy for the long term, but they don't need to store a lot, and so they store less energized forms of these triglycerides, and they're in the form of oil. Okay, we get vegetable oil from vegetables. We get canola oil from canola. You get safflower and sunflower seed oil. You get olive oil. all of those oils that you can buy in a store come from plants. Okay, and those plants store the energy for a long term in the form of that oil. But again, it's liquid at room temp, so it's healthier for you. So the other type of lipid, and probably the most important type of lipid when we talk about biology, is this phospholipid. Okay, and if we break that word down, phospho means phosphate, and we have a phosphate head, and lipid means that it's a lipid. So it's a lipid that contains a phosphate head, and it's different than a triglyceride because it only has 2 fatty acid tails. And remember, we've talked about structure dictates function a lot. Well, structure dictates function in lipids also. What is the purpose of a triglyceride? Remember, the triglyceride looks like this. It's a glycerol head with 3 fatty acid tails, and the purpose of the triglyceride is to store energy long term. Where does it store the energy? In the tails. It makes sense that a lipid that is structured to store energy should have 3 tails. And a lipid that isn't structured to store energy, such as the phospholipid, should only have 2 tails because it doesn't store energy, right? This is the lipid that makes up all bilayers, okay, or makes up all cellular membranes. Phospholipid bilayers, okay, phospholipid bilayer is 2 layers of lipids, and these lipids have a hydrophilic or polar head, and they have a hydrophobic or nonpolar tails. Now, we've already talked about in class what polarity means and what nonpolar means. Okay, polar means that, like water molecules, you have an unequal distribution of charges, and nonpolar means that you don't have an unequal distribution of charges. And so these heads are going to interact with water, and the tails are not going to interact with water, which leads me to my next picture, which is a phospholipid bilayer. Okay, notice these little pink kind of jellyfish-looking things? Those are the phospholipids. Now, a phospholipid like this one looks just like we drew it. It has a phosphate head. It has 2 fatty acid tails or chains. Notice that all the heads are towards the outside, and all the heads are towards the inside of the cell. And because the tails point towards the inside of this membrane, this produces this zone of hydrophobicity. Remember, the tails are hydrophobic, meaning they fear water. The heads are hydrophilic, meaning they love water. And so we have H2O out here, and we have H2O in the cell. Okay, and so the only place where we don't have water is right through this middle of the membrane. And that brings in why we have protein channels. Okay, because when things like water want to flow through the membrane, they're going to flow through these things called protein channels so that the water doesn't actually touch these tails. Because again, the tails are hydrophobic, do not like water. They do not interact with water. Okay, some of the things you're going to see within the cell membrane are proteins, carbohydrates, and cholesterol. These chains right here are the carbohydrates. We call these glycoproteins. They are proteins that have carbohydrate chains attached to them. These would be a protein, but more integral protein. Okay, they don't have the protein or they don't have the carbohydrate chains attached to them. So these would be glycoproteins. These would be integral proteins. And then you have channel proteins, and again, a channel protein sounds like what it is. It's a channel or a tube that connects the outside of a membrane to the inside of the membrane. One of the most important parts of this bilayer are these little molecules here, and those are cholesterol. And cholesterol adds the structural integrity to the membrane. If you think about a brick wall, bricks are not just stacked on top of each other, they're actually cemented to each other with a layer of mortar between them. Okay, and so these cholesterol molecules are exactly what that mortar is in a brick wall. They add the structural integrity to the membrane. The membrane would not be as strong as it is without the cholesterol. Okay, phospholipids create the actual barrier. Proteins allow the membrane or cell to communicate with other cells and bring materials in and out. The cholesterol holds the membrane together. Okay, so that's it for lipids. Remember, we have triglycerides and phospholipids. Both of them do something incredibly different. Phospholipids create a membrane which allows us to have a barrier between the inside and outside of a cell. Triglycerides are our long-term energy storage molecule, and they differ in structure. Remember, animals have triglycerides that are more saturated with hydrogen. Plants have triglycerides that are more unsaturated with hydrogen. The saturated fats, remember, are solid at room temp and are typically bad for you because of that reason. The unsaturated fats are liquid at room temperature and are more healthy for that reason. But make sure you took good notes. Make sure you come to class with questions. Make sure you understand the difference between phospholipids and triglycerides. And next lecture, we'll talk about proteins. Mr. P, out.